right, so good morning, good afternoon, everyone. Welcome to another broadcast with us here at Exploring by the Seat of Your Pants. I know we've got some new faces in the crowd today, and so if you are new to us, we're all about bringing conservation, adventure, and science into classrooms around the world. Now, everything we do goes to our YouTube channel live. There's a bunch of you there right now, so a big welcome to our YouTube audience as well, and stays there forever. So if you want to watch this program in 3 weeks or 3 years or catch any of our other Gairdner Foundation series this week and beyond, I really encourage you to head there. There's so much to keep the learning going, over 3,000 broadcasts since 2014 alone. Now, today, as I mentioned, we are continuing our epic program series in conjunction with the amazing folks at the Gairdner Foundation. So every year, we get to bring in some of their incredible laureates featuring the very best biomedical and health researchers on planet Earth. For the last 2 days, we've had the chance to hang out with Dr. Bonnie Bassler, talking about how bacteria talk with one another. We wrapped up earlier today with Chris Musquash, talking about Indigenous-led mental health initiatives in Ontario and beyond. And we've got CRISPR and cancer and all sorts of other great stuff coming up in the days to come as well. But I'm really thrilled to welcome back Dr. Anna Blakney. She joined us last year for a really exciting program. She's got the biggest audience of our crowd today, of our Gairdner group. Don't tell the others. But it's really exciting to have her back as she gets to talk about next generation vaccines, RNA therapies, and more. A lot of us are very familiar with these terms after this pandemic that happened over the last few years. I'm going to turn it over to Dr. Blakney to tell you a little bit about her amazing work, not just doing research, but also sharing that work with the public. Some of you might well know her from TikTok, over 4 million likes, communicating really complex scientific topics to a really wide audience. So always a thrill to have her back, Dr. Blakney. Thank you so much for joining us today, and welcome to Exploring by the Seat of Your Pants.
Thanks so much for having me. I really love doing this type of stuff. So I was very delighted to be welcomed back. And I will not tell anybody that there is a bigger audience today, but I'm very flattered. So this is really fun. Let's get going. Let me just share my screen.
Yes, while you're doing that too, I'll note for our live audience, YouTube audience, I know you're high schoolers, so sometimes a little reluctant to ask questions, but lots of opportunity for Q&A at the end. We'd really like to have a conversation with you guys, so please do feel free to not be shy. And Anna, we are good to go. This looks fantastic. Take us away.
Excellent. Well, yeah, thanks so much. So I am an assistant professor here at the University of British Columbia in the Michael Smith Labs in the School of Biomedical Engineering. And today I'll walk you through where I think this technology is going for next generation RNA vaccines and therapies. So just to give you an outline, I always think it's really fascinating to hear about how people arrive at their career. I think as a student, that's one of the most kind of nebulous things. And so I thought I would share how I got here and then talk a little bit about the research that we're doing here in my lab at UBC, and then take some time to speculate on the future of RNA medicine. So just a little introduction to me. I'm actually originally from Colorado. I did my undergraduate degree there at the University of Colorado in chemical engineering. So I'm actually an engineer by training. I then went on to do my PhD in bioengineering at the University of Washington in Seattle, so just down the road here from Vancouver. I then took a little bit of a left turn and did my postdoc in a pure immunology lab at Imperial College London. And then obviously now I'm at UBC in the School of Biomedical Engineering and the Michael Smith Lab. So one thing I just really wanted to highlight at the beginning of this presentation is, to me, science is really like a passport. So growing up in Colorado, which is a fabulous place to live, but I always thought I'd end up in Colorado. I didn't really have much ambition to leave, but science has really made me realize like there's a whole world out there, and science is a really interesting way to do that because you get to experience just views and perspectives from people from all different parts of the world. So some of the places that I've ended up through my scientific career, lots of really beautiful, interesting cities to live in, like Boston, Pittsburgh. I did part of my PhD at the University of Cape Town in South Africa, obviously Seattle, London, and Vancouver. And so this is something that I really wanted to reiterate to you all today, is that there are really fantastic opportunities in science if you're looking for a way to kind of get a more diverse experience. I also wanted to touch on, I talked a little bit in my introduction about this idea of immunology and engineering coming together, and people never know what this term means, even when I talk to like other people in immunology or engineering. So I thought I would just clarify kind of what this means to me. So my background is in chemical engineering, and I think what most people think of that, other than that sounds very boring or very intense, is kind of the scale up and production of different processes, really. So typically chemical engineers went into like oil and gas, so working at refineries, which is one of the things I could have been doing. But I was really interested in combining this with immunology. So how do we engineer the immune system and use this as a way to make new vaccines and therapies? So this is where we come into immunoengineering. So what I think about it is really applying these engineering approaches. So having kind of these design criterias and the engineering design process, but using that in the context of immunology so that we can improve human health. So to go over the research that we do here in my lab at UBC, it's been a very exciting time to be in the field in the past few years. I remember prior to the pandemic, I would start every single talk I gave with, probably half the lecture on just telling people what RNA vaccines were and like trying to convince them that they could work one day. So obviously now that billions of people have had RNA vaccines, the plane is off the ground. And so it's really exciting to think about where do we go from here. So my lab at UBC really has kind of three different aspects to it. So we work on RNA molecular engineering, which is just a fancy way of saying, how do we design RNA to express protein for longer or in certain cell types? We also work on biomaterials design. So some of you might recognize this as a lipid nanoparticle, which is one of the technologies that was developed here at UBC that had a really great impact on the COVID-19 vaccines. So how do we get these nanoparticles to the cells and tissues where we want our RNA cargo to go? And then really thinking about the frontier of RNA medicine. So how do we make better vaccines? How do we use this for other applications? And really think about how do we make the most of this technology that's been developed in the past few decades. So I'd like to start off by thinking, why do we even need new types of vaccines? Why is this still part of the work that we do? There's a number of reasons, and I've just covered a few here today. So we still have lack of efficacious treatment for many devastating infections. So for example, HIV, that's a virus that's been circulating for many years now that we don't have a vaccine for yet. So we need to think about new approaches for that. In recent years, we've also seen the emergence of multi-drug resistant bacteria. So vaccines could be a way of combating this. We're always looking to improve the safety profile of already licensed vaccines. One that we can all relate to now is disease outbreaks. So if there's a new pathogen, we'll need to develop a new vaccine for that specifically. We also need to consider the changing age structure of the population. So as people are living longer, your immune system changes as you get older. And so we really need to think about different strategies to vaccinate the elderly as well. And of course, we're always interested in reducing the cost and time that's required to produce vaccines. So here I have a picture of how we still make the flu vaccine in eggs, which, as you can imagine, is very time and resource intensive. So there's lots of areas where we can really make a positive difference here. So for those of you who aren't familiar, I just wanted to walk you through how you make an RNA vaccine against a brand new pathogen. So this is actually an incredibly fast process to do because this is what we call a platform technology. So the way we do this is a picture of the SARS-CoV-2 virus, and this pink lollipop-looking structure on the surface of it is the spike glycoprotein. So this is just a protein on the surface of the virus that is actually what the virus uses to enter cells. So we take the RNA code for that protein and put it into our RNA. We then encapsulate that into lipid nanoparticles. So these are kind of like little lipid particles that are able to guide the RNA into cells. And then we inject that, usually starting in mice in our preclinical models, but then obviously advancing to human clinical trials. So one of the things that was really remarkable during the pandemic is that we saw a really high efficacy, especially initially, of the mRNA vaccines against COVID-19. So this is one of the graphs from the clinical trial data that Pfizer and BioNTech published. And this is looking at the incidence of infection. And so the orange line is the Pfizer and BioNTech vaccine, and then the blue is the placebo. So you can see that after a certain point, there was a very low increase in the number of cases of COVID when you got the vaccine, whereas for the placebo, this continued to go up over time. So when these data were initially released, there was a 95% protection against COVID-19, which is actually a really epic efficacy to achieve for a vaccine. Most vaccines are not that high. And another really important part of this is the safety profile of these vaccines, which they said was over a medium of 2 months, similar to that of other viral vaccines. So this is to say, there are some side effects from these vaccines, but some of those side effects are actually adding to the immune response that you're seeing. You will never have a vaccine that has absolutely no side effect. And so this is a really great safety and efficacy profile. So what are the challenges with RNA vaccines today? I thought I would walk you through some more clinical trial data, but stick with me. I'll walk you through what all of this stuff means. But I really want to convey that one of the biggest, emphases in the field right now is minimizing the dose of RNA so that we can reduce the number of side effects for patients. So this is some of Moderna's phase 1 clinical trial data. So in this clinical trial, they were aiming to just look at the safety and side effects that they saw in patients. And in order to do this, they tested out 3 different doses of their RNA vaccine, so 25, 100, and 250 micrograms. And they categorized the side effects as mild, moderate, or severe. And what I think is just very visually apparent from this data is that as you increase the dose of RNA, you increase both the frequency and the severity of side effects. So we know now from all of this clinical trial data that the dose of RNA is directly correlated to the frequency and severity of these side effects. And so really one thing for us as scientists that's so important is really working on minimizing the dose of RNA for non-pandemic contexts, such as if people want to get a seasonal influenza virus, as well as other applications of RNA, they're not going to want to deal with those side effects. I just got my COVID vaccine, the new one, on Friday, and I can attest, I was thinking the whole time that I had a fever and side effects from that we do need better RNA vaccines. So this is an ongoing aspect of our research as well. So one of the ways that my lab does this is by using a type of RNA called self-amplifying RNA. So this is actually just a type of messenger RNA that's actually able to make copies of itself once it gets into a cell. So I have a simplified schematic here that I'll walk you through. So on the right, we have messenger RNA, like what's in the COVID-19 vaccines. Once a copy of that RNA enters a cell, it engages with the ribosome and you get translation of some of your vaccine antigen. So this is what you make an immune response against. So our self-amplifying RNA starts out very similarly. We get one copy of the RNA into a cell. It engages with the ribosome and starts making protein from that RNA strand. We do get some translation of our antigen, but more importantly, it makes a replicase enzyme. So what a replicase does, as the name implies, is it replicates the RNA. So it makes exact copies of this original strand of the saRNA. And for the purposes of this diagram, I just have 3 copies here, but it's actually thousands of copies within the cell. And so because of this, you get more translation of your antigen. You get a higher amount of protein expressed from this initial dose. And we're usually able to use about 100 times lower dose of saRNA compared to messenger RNA. So enter COVID-19. How did we actually go about making the vaccines against this, and where do we kind of go from here? So as a postdoc, before I started my lab here at UBC, I was part of a team at Imperial College that developed the first in human saRNA vaccine against COVID-19. So I wanted to walk you through the steps of how we actually did that as an academic team. So the circulating strain in 2020 was first sequenced and made publicly available on the 10th of January. Our team accessed the sequence on the 20th of January. I very coincidentally flew to Vancouver the next day to interview for my current position on the 21st of January. So as you can imagine, this was a very busy time. We then ordered the DNA oligos on the 22nd of January. We then assembled our plasmid on the 28th of January. We confirmed the appropriate expression from our construct on the 12th of February. and started our studies, preclinical studies in mice on the 13th of February. This was a first in human trial, and so in order to do that first study, we had to do a toxicology study in rats that we started on the 30th of March. While we were waiting for those results, we submitted our paper on this preclinical work and then started our clinical trials in humans on the 19th of June. So even as an academic team, we were able to go from a brand new virus in January and achieve starting our clinical trial within just 6 months. So this platform technology of like it really being easy to make and test these new RNA vaccines has just really sped up how easy it is to make new vaccines. It doesn't change anything about the clinical trials or how we test them. It's just easier to produce them because you do it in the same way for every RNA vaccine. And I'll get into that in a bit. So I also wanted to highlight that saRNA technology is really taking off. It's very exciting. So there's now an approved saRNA vaccine against COVID-19. This was approved in India, and it was developed by a company in Seattle called HTT Bio. So their vaccine was approved at a dose of 5 micrograms compared to the 30 or 50 micrograms that are used in the Pfizer and BioNTech or the Moderna vaccines. So it's really exciting to see this technology being more widely used and now approved in humans, which was a huge milestone, really. So I wanted to talk to you all about how we actually make vaccines and why this is such an important component of mRNA and really the overall potential for this technology. So the way that we make kind of old types of vaccines, so protein and viral vaccines, one, I have a picture of how we made the flu vaccine in 1957. So this is a vaccine that's manufactured in eggs. And I have a picture down here from 2021 of how we still manufacture the flu vaccine. So it's again still made in eggs. This is part of the reason why we in February each year have to decide what strains are going to be put into the flu vaccine, so for the Northern Hemisphere at least, so that it's available in October. And so this is, really one of the reasons why the flu vaccine typically doesn't have a super high efficacy, but we think RNA vaccines can improve that. So for other vaccines, we use these huge bioreactors, which grow cells, which then either make the proteins or the viruses for the vaccines that we need to make. As you can imagine, this is like a huge manufacturing challenge. You have to have these big factories with lots of bioreactors and then purify out the protein or the virus. And again, it's very time and resource intensive. On the other hand, for RNA vaccines, they're made completely synthetically. So just means we put all of the ingredients into a small bioreactor. And just to put it into perspective, in a 100 mil volume, which is about the size of a cup of coffee, we can make about 1 million doses of vaccine. So even in my lab here at UBC, we're able to manufacture 1 million doses of RNA vaccine in a very reasonable time frame. So the reason I think this is a really impactful technology, and just to put it into perspective, I like to ask the question, this is kind of a thought experiment for everybody. We're going to have to do a little math. I'm not great at mental math, so I have it all spelled out here on the slides. But I want to ask you, how many Olympic-sized swimming pools would it take to vaccinate the entire world with an RNA vaccine? So I did these calculations specifically for the Pfizer and BioNTech vaccine. So if we start with the population of the world, which is 7.7 billion people, everybody initially required 2 doses. So that's a total of 15.4 billion doses. Each dose is a volume of 0.3 milliliters. That means our total required volume is 4.62 million liters. So if we break that down into an Olympic-sized pool, each Olympic pool is about 2.5 million liters. So the answer is we need about 2 Olympic-sized swimming pools. And while it seems like a huge volume of liquid, and it really is, the thing about it is it's very feasible, right? So we've already seen that we've been able to vaccinate billions of people with an mRNA vaccine. And that's one of the reasons why the mRNA vaccines were so successful during COVID, is because it was much easier to scale these up than any other type of vaccine. So I want to talk to you now about a topic that I think an initial take on this slide sounds very boring, which is the future manufacture of vaccines. But I'm going to try to convince you that this is actually a really exciting aspect of this technology. So for the clinical trial that I worked on in London, I wanted to walk you through about how we actually made our vaccine. And this was one of the biggest challenges that we had for this clinical trial, actually. So our RNA came from a company called TriLink in California. Our lipids came from here in Vancouver from Acuitas Therapeutics. These were shipped to Austria to Polymoon to be put into the formulation, into the lipid nanoparticles. That formulation was then shipped to Italy to be put into vials. And then those vials were then shipped back to the UK to start our clinical trial. So even at the time, none of this vaccine was made in the UK. And this wasn't only a challenge that the UK had. There were many countries all over the world that weren't able to manufacture these vaccines themselves. But I think that's going to change. So instead of having these like large centralized manufacturing centers, I think what we'll see in the future is actually many smaller regional manufacturing centers where we can make whatever that community requires. So whether this is a COVID vaccine or a seasonal flu vaccine, or even a personalized cancer vaccine, they can produce those all in the same setup and really serve the population and whatever they need. So one of the things I also really wanted to highlight is, RNA has been used for COVID vaccines, but I think there's so much potential and things we can apply this to. So this is actually not my slide. I took this from BioNTech's website, but I think it does a great job of summing up kind of where we are and where we're going in the future. So today we have the Pfizer and BioNTech vaccine. Tomorrow, I guess, in the pipeline, things that BioNTech is already working on. So mRNA vaccines for other infectious diseases. They've had a long history of making mRNA cancer vaccines. They've also have these CAR T cell amplifying mRNA vaccines, as well as systemic immunotherapies, and then are starting to think about other applications such as autoimmune diseases, allergy, inflammation, and regenerative medicine. So I think we've just started to see what this technology can do. And there's still a lot of optimization and science that needs to be done, but it's a really exciting time to see how we can continue to positively impact human health. So one of the things I realized during the pandemic, and I wanted to cover today, is really how important science communication is. So something that was like very obvious to me during the pandemic is, as scientists, we think everything we work on is great and that everybody will just be like jumping, trying to get a vaccine. But we need to be realistic, right? So at the end of the day, if nobody wants to take the vaccine, there's no point in us making it. So the communication and education is a very important aspect of what we do as scientists. So on the right, I have a graph that was published by Heidi Larson, who studies vaccine hesitancy all over the world. The top schematic is a representation that says vaccines are safe. The middle is vaccines are important, and the bottom is vaccines are effective. And the grading scale is the more blue it is participants strongly agree, whereas the more orange it is are patients or people who do not strongly agree. And what I think is really interesting takeaway from this data is that it's actually largely blue all over the world saying that vaccines are important. But there seems to be a disparity between people strongly agreeing that vaccines are both safe and effective. And so I'm really interested in kind of just understanding, where does this vaccine hesitancy stem from, and what is the missing link between believing that vaccines are important but not safe or effective, which is so far from what we have exactly what the data indicates. So this is why I think public engagement is really important and an integral part of what we do as scientists. So what I realized during the pandemic is that the pace of science is just not matched by the pace of public communication. And to exemplify this, if you think back to January 2020, there were just a lot of scientists that didn't even know what an RNA vaccine was. So many people, even in the field, hadn't really heard of them or weren't really familiar with the technology. And now billions of people have gotten them. So of course, there's lots of questions that people had about this technology and the development and how it had been studied and the long-term safety profile. And as scientists, we need to be able to answer those questions. So one of the things I did during the pandemic is I started a TikTok platform channel that's dedicated to educating people about RNA vaccines. So I actually started this in October 2020, kind of right before the vaccines were starting to be available. I was recruited to an organization called Team Halo, which was started as a collaboration actually between the United Nations and the Vaccine Confidence Project, which is run by Heidi Larson, the woman who studies the vaccine hesitancy. And their whole goal was to get scientists and clinicians on TikTok to just answer people's questions and educate them about the vaccines that were being developed, so that there's really kind of no middleman between the people that are actually doing this work and people who have questions. So I think TikTok it's not just a platform for dancing teenagers, although that's maybe how it was initially understood or branded, but I think it's actually a very elegant platform to show people exactly how we make and test vaccines, and we can also then directly confront misinformation or questions that people have. I think something about it that really works is that the videos are short and engaging and offer a palatable amount of information. It's like 15 or 30 seconds. And so you really have to think about the message that you want to get across, because you don't always get opportunities like today where I can kind of go more in depth and tell everybody about kind of where we've come from and where the development has happened. So one of the really fun aspects of this for me is really being able to highlight and recognize the scientists that were behind the COVID-19 vaccines. So I had a number of people on my channel, but one thing I wanted to highlight was some of the guests that I was just really pleased to have. So one of whom is Blakney, who is a professor at MIT and also a co-founder of Moderna. And the other in the middle here is Professor Peter Kullitz. So he's a professor here at UBC. He was co-founder of Acuitas Therapeutics, which is the company that really came up with the lipid nanoparticle technology that formulated the Pfizer and BioNTech vaccine. And if you'll notice in the comments over here, this is so sweet, the things people are saying. You get a lot of trolls and negativity on social media, but I thought this was just really, really sweet. So you see people saying like, thank you for saving the world, real life superhero. I would love to get it. Thank you for saving lives. So grateful for you, Peter. This is so wholesome. So what it made me realize is, people really appreciate scientists, but they also just have no idea who they are. And so part of my goal is to make more scientists household names. So to sum everything up, I just wanted to walk you through everything again. So for RNA vaccines, we've seen, the mRNA vaccines have really enabled a rapid response to the SARS-CoV-2 pandemic. Why the manufacturing, I think, is going to be such a game changer for this technology is that mRNA vaccines are made synthetically and require a much smaller footprint and time frame to manufacture than traditional vaccines. We've seen that the side effects have been shown to be proportional to the dose of RNA, so we need to minimize the dose so that we can get rid of some of these side effects. And on the science communication aspect, I think communicating to the public about science is imperative. So I don't think this is just true specifically for vaccines. I think this is for every realm of science. And I think as researchers, we need to do a better job of this. So I'd like to acknowledge everybody here at UBC. That's a part of my lab, as well as our lab alumni and our funding sources that fund this work. And with that, I will take any questions.
Fantastic. Well, thank you so much, Anna, and for highlighting your lab at the end. It's been something we've seen in all our Gairdner programs and just increasingly lately, just highlighting the collaborative nature of science, how many people are involved in discovery. It is very seldom a lone genius working alone. In fact, probably never a lone genius working alone. You're always building on the work of other people, always collaborating, learning from colleagues. So really, really appreciate that. Also, I want to go to Boston now from that picture at the beginning. YouTubers, live class, Mr. Stim's class. If you guys have questions, I will come to you in just a second. YouTubers, please feel free to chime in. We had them written in while you were chatting, and I want to highlight and sort of get underway with them, give our rest of our audience a second to think about this. So our first one is, we had this earlier in a couple programs ago with bacterial research and vaccines as well. So I'm curious how AI might be helping this. This is a very big buzzword nowadays. I'm sure you're getting this question a lot. Is artificial intelligence affecting your research at all, speeding it up, no impact whatsoever? What's the deal?
For sure. So AI is not something we specifically work on, but likely will incorporate into kind of our pipeline in the future. So how this helps us as RNA researchers is, in your RNA code, there's actually so much we still have to understand about kind of the structure of the RNA and its downstream function. And so this is really a great task for AI because we can, as we start using different designs and testing it for vaccines or therapies, we can feed this into a big data model that can then help us design better RNA in the future. So this work is really just starting to come online, but I think will make a big difference for RNA technology as well.
Very cool. It's a really interesting time in sort of this world of science and biomedicine. You were talking about AlphaFold a couple of programs ago, and so there's a lot of big changes happening really rapidly. So if you're interested in this field, our grade 12 students today, there's pretty much infinite realm for discovery in the next few years. It's been so exciting to see that pace of vaccine development as well, which leads us to our next question, actually. So, in COVID, it was this global effort. It was scientists all over the world. It was every lab, every government working on making this a possibility and a reality. How long does a vaccine typically take to develop? And this includes sort of new mRNA vaccines. What's the deal?
Yeah, that's a great question. So I think, if you ask people this prior to the pandemic, they would say it typically takes about 10 years to develop a new vaccine candidate and get it approved. This has a little bit changed now. So obviously during the pandemic, it was such a different time because the whole world, scientists, clinicians, funders, regulatory bodies were all focused on one disease. And it was actually, to me, a really interesting kind of feat of humanity and teamwork and cooperation, how quickly we were able to move these through. Now that we're back to kind of a steady state, I don't think the speed will be quite the same because it's also just not as urgent, I would say. But what we're seeing now for RNA is a little bit different. So you may have seen that, there's updates to the COVID vaccines every year now. And these vaccines don't have to go through a huge phase 3 clinical trial when it's just a small change. They're just changing 1 or 2 bases to basically match the mutation of the virus. So this is similar to the way that the flu vaccine is released every year, right? Like we don't have to do huge clinical trials because we know like the platform is safe and just these minor changes don't make a huge difference for that. So I guess it depends on what the vaccine is as to how long it will take, but I'm hoping that it will minimize the time in a obviously like safe and careful way to develop new RNA vaccines, given that they're somewhat similar to the COVID vaccines that have already been developed. So I think it's changing. Of course, our first and foremost efforts are always about safety, but I think we can make this so that these medicines are available more quickly to really help people.
Yeah, it's really fascinating getting to see sort of Nature and Science publications, sort of the 2 gold standard publications for up and coming science, and how much is on, incredible vaccine developments. It's a really exciting time. I get to say that for a lot of different themes that we bring on Exploring by the Seat of Your Pants, but biomedicine right now is just skyrocketing, which is very exciting to see.
Yeah, that's right.
Speaking of trials, and this is a great follow-up question, how many people are involved in a trial and how do you get involved? And one of our students or teachers wanted to be a part of a trial, how does that actually look? Or can you volunteer to be part of that? What's going on?
Definitely. So usually those trials are run either like at or by researchers at a university. So a clinician will run those trials, or big pharma companies will also have things in development. So I actually think that's I consider that an important part of, we all benefit from these medicines. And I think it's really important to be involved in that testing as well. So as far as the vaccines, usually they're so they're all listed on a website called clinicaltrials.gov. That's you can find them in your area and see if you're eligible. I think it's a really great thing to volunteer for and contribute to, obviously, because those patients are super, super important. So that's the main way. Otherwise, sometimes locally here at UBC, I'll see advertisements for it, but it's very much depending on kind of where you live and what research is being done there for sure.
Very cool. Thank you, Anna. We're going to head to Mr. Tim's class, our Careers 12 crew. If you guys have questions live, I will come to you. Go for it and take us away if you have any queries. Hi, folks. Go for it. Yeah, hi.
So the self-replicating RNA that you the self-replicating RNA that you use instead of the regular RNA in the vaccines that helps lessen the side effects by injecting less RNA, is that decreasing the efficiency of the vaccine, and will you need more doses for that?
Cool.
Yeah, that's a really good question. So I just to make sure I'm on the same page. So your question was whether using like a lower dose of the saRNA compared to the mRNA vaccines, does this decrease the efficacy of it? Is that right?
Yeah.
Yeah.
So, yeah. Perfect.
Well, so yeah, so really no. So really what we see is by using about 100 times lower dose of the saRNA compared to mRNA, we see an about equivalent immune response. And so the reason for that is that even though we're using a lower initial dose of the saRNA, it's just amplifying in certain cells. And so we're seeing about the same amount of antigen there from both vaccines, but we can just give you a lower amount of RNA to begin with. But the immune responses should be equivalent.
Okay. A great follow-up question from our YouTube chat on the saRNA. So what stops it from replicating? So it starts replicating, cool, we get a bigger impact. Is there like a kill switch mechanism that stops from going crazy? For anyone who wants to know just as much as you can explain about this.
Yeah, that's a great question. It's something I get asked often as well. So what controls the replication is just the normal, basically the mechanisms in your cell that control a normal viral replication. So at some point, the decay rate of the RNA overcomes the replication rate, and you see cessation of the replication and the protein expression. So in the same way that like a virus may replicate for some time, but then finishes, it's the same for the saRNA. It's those exact same mechanisms. So there's not necessarily an engineered kill switch in it, but we can control the amount of time that it replicates for, and sometimes in specific cells as well, just by the design of the RNA.
Very cool. I highlighted this during one of our programs a couple days ago. Just a great primer for our grade 12 audience, university audience. Immune by Phil Detmer, who does an amazing YouTube channel on SciComm, is just a spectacular book. It is insanely complex science. Like cell biology is ridiculous. If you really get into the nitty gritty of how cells work, it's mind blowing. I think that's a good primer for a lot of people and might lead some future inquiry if you're keen to follow up. Mr. Sims class, I'm going to head back to you guys. We'll take a couple more. Time flies and you're having fun at us. So you've got time for more questions. But Careers 12, if you got another, come on up.
So I'm curious, what stops the replicase enzyme from replicating other RNAs in the cell?
Mm-hmm. Great question. And actually, right before this, I was giving a presentation to the Dean of the Faculty of Medicine here and his International Advisory Council, and they asked me the exact same question. So thank you for the very sophisticated and an insightful question. So what stops it from just amplifying any old mRNA that's in your cell is it's very specific to the sequence at the beginning and end of the RNA. So if you change those sequences at all, it will not replicate. And it also means that it can't just replicate a typical mRNA that you might find in cells. So it's very, very specific.
Very cool. Great question, Mr. Sims. All right, I'm going to wrap up with a couple more questions. One is just to say, is there any resource or other person that people should follow, like a great go-to source for great information up to date on the work of this field in general? And this could be social media, this could be a news platform, this could be an individual, whatever you want to share, we'd love to hear it.
Yeah, I think actually one of my favorite people on social media to follow is Science Sam. I think she does a great job, very broad, but she does a great job of just summing up in very kind of palatable way of understanding many different sides of science. So she's a great follow if you're interested in learning more.
Fantastic. We've had Sam on the broadcast. I sort of came up in Toronto with her in the early days of major SciComm in the city. So does spectacular work. Can't recommend her enough. Thank you for that, Anna. How nice. And then just as a final question, is there any part of your job specifically that you are most excited about? Like something that gets you up in the morning that you can't wait to do personally or in the field in the next couple of years?
Yeah, I think to me, what's so exciting about science and kind of what I find very empowering about it is we have an idea we can just kind of do it and perform it. So one of the things, maybe an example of this I'll give is, I got my COVID and flu vaccines on Friday. And prior to getting them, I was having a debate with my lab and other colleagues of, should I get them in the same arm or different arms? And there was a lot of debate about like what was better for your immune response. And one of my colleagues was saying, well, like, oh, I had to play tennis the next day, so I didn't want to have 2 sore arms. So I just got them in one arm, which I feel like is probably what most people want to do. But in my mind, I was like, oh, but it probably makes more sense to like do it in like one in one arm, so that if you have any side effects to one, but also then they drain to different lymph nodes and you probably get a better immune response. And so I was thinking about this and talking to my lab, and I was like, we should just test this. Like, let's find out if it's better to get it in different arms or one arm. And that's what I think is so empowering about like being a researcher and in science is you can have these ideas, but then you actually have the power to like answer the question yourself.
Very, very cool. I just got my shots a couple weeks ago. Students, go get your shots if you haven't got a flu and COVID vaccine. It's been funny talking with people over the last few weeks because people are like, oh yeah, COVID and flu. And it's like flu is a historical major killer. And just, who wants a week off school or work? Or maybe you do want a week off school or work, but hopefully not. Think of those around you and get your flu vaccine. Anna, this has been so much fun. Thank you so much for joining us. Again, I want to highlight, if people want to check you out on TikTok and beyond, you're really easy to find, lots of great videos there. And then, of course, this is all part of our Gairdner series. So check out Gairdner.org, all the amazing laureates they feature in a wide range of biomedical and health programs. We've got a few more coming up to wrap up the week, so do stay tuned for that. And for now, I'll just bring in Mr. Sims' class to say thank you and farewell as well. And Anna, have a wonderful rest of your day. Bye for now, everyone. Thank you so much.